Hello and welcome. We are pleased to have people joining us from all over the globe to take part in a conversation about treatment and research in progressive MS, a disease which impacts the lives of millions. During this webcast, you'll meet Dr. Mahad and Dr. Zalk, leading MS researchers who are working to build new understanding about MS progression in order to develop new effective treatments. You'll also hear from Dr. Zaratin, a member of the Scientific Steering Committee of the International Progressive MS Alliance. This committee is comprised of leading international scientists and is charged with developing a scientific roadmap to develop new treatments. It promises to be an informative and inspiring 30 minutes. So if you're like me, grab pen and paper so that you can jot down notes because you'll want to remember this information. My name is Angela White, and I am joining you from Washington, D.C. in the U.S. I have been a volunteer with the National Multiple Sclerosis Society since my diagnosis of multiple sclerosis in 2002. I serve in a variety of volunteer roles at the Society. As an MS activist, I work to ensure the voices and needs of people with MS are heard by policymakers at all levels of government. I also serve on the Black MS Community Engagement Advisory Committee, where we ensure all members of the MS community feel seen, heard, understood, and valued. And the Community Review of MS Research Committee, where we review MS research applications for the Society to ensure they achieve the greatest impact on improving the lives of those living with or affected by MS. I also participate in the MS International Federation's Patient-Reported Outcomes Initiative, helping improve the effectiveness of MS research worldwide and ensuring that patients' concerns and their outcomes are an inclusive part of the entire process. In 1 minute, I will introduce you to our outstanding MS experts. But first, I'd like to recognize our webcast sponsor and share some information about the International Progressive MS Alliance. We are so grateful for the generous support of our sponsor, Sanofi. Thank you for helping make this webcast possible. The Alliance is an unprecedented global collaboration to end progressive MS. It includes 19 MS organizations, hundreds of researchers and health professionals, the pharmaceutical industry, companies, foundations, donors, and people affected by progressive MS. In short, the Alliance has brought the world together to solve progressive MS. To date, the Alliance has invested over 24 million euros in research with an overall commitment of 60 million euros through 2025. And if you are watching this webcast, you likely know that ending progressive MS is an urgent and unmet need. That is why I am so pleased that there are with me today 3 leading MS experts who are working to fuel research to develop new, life-changing treatments. I'm going to begin with Dr. Zaratin. She is the Director of Scientific Research at the Italian MS Society, and as I noted earlier, a member of the Progressive MS Alliance Scientific Steering Committee. Dr. Zaratin, people with progressive MS are deeply frustrated at the lack of treatments that can effectively slow progression. With so much progress having been achieved in developing drugs that address the inflammatory aspects of MS, little has been accomplished in treatments that slow or stop progression. What do you think stands in our way of this?
Thank you for the question, and good morning, good afternoon, everybody. It's a pleasure to be with you today. So let me start answering the question by saying that there are at least two main challenges in developing therapy for progressive MS. So in particular, we don't know what is broken in progressive MS. At the level of biology, we don't know what is really driving the gradual little by little progression. And of course, if we don't know what is causing it, it becomes very difficult to find a solution, even if not impossible. Let me say that it's like when your car breaks down, and you don't know whether it is because of engine, because of the wheels, because of the batteries. And if you don't know where things are broken, it's going to be very difficult to fix it. So this is why one important research area is to understand the mechanisms that drive progression. The second challenge is related to the difficulty to test potential therapy for progressive MS in a quick and effective fashion. Let me give you the example of the relapsing MS. In relapsing MS, you can measure, look at the lesion on MRI. And if you have a drug, you can test the drugs and see whether it's able to reduce the lesions. And if it does, then you can test the drugs in a larger trial to see whether it's able to reduce the number of relapses. So in a short MRI trial, you're likely to identify the drugs that can be the winner in the larger trial. So short MRI study, a large trial that help you to pick up the winner. This is not going to be the case for progressive MS. We don't know how to design a proof of concept trial. We don't have a biomarker. We don't know a biomarker that can help us to pick up the winner. And therefore, it is very difficult to develop therapies for progressive MS. Again, not impossible. This is why this is the second important research area that, as we're going to discuss likely later, this is the focus of Allianz research portfolio.
So, Dr. Zaritan, how is the Alliance working to overcome these challenges?
Yeah, so exactly as I was saying before, Allianz is driving multiple areas of research to be able to tackle these challenges from different perspectives. But one important key investment that Allianz has made recently is funding 17 challenge hours. These challenge hours are very innovative research studies. And the goal, the objective of these research studies is exactly to understand the mechanisms that drive progression. And not only this, is also about to identify novel therapeutic targets, and why not some initial molecule that can modulate this mechanism. The Allianz Challenge Awards are the results of a worldwide call for proposal application, and each awards is was expected to be innovative and collaborative as much as possible, but in particularly be innovative in a way to shift the paradigm to help us to identify what we have not discovered so far in terms of understanding mechanisms that drive progression.
I'm delighted to hear about the emphasis on collaboration. So thank you for sharing that. And it is so encouraging to think that these projects could unlock the door to understanding MS progression. I'm also delighted that we have two of the lead investigators who have received a Challenges in Progressive MS award with us today, Dr. Mahad and Dr. Zalk. Dr. Mahad is a senior clinical lecturer and honorary consultant neurologist at the University of Edinburgh in Scotland. His research is focused on bioenergetic changes in progressive MS. Patients, also in particular, the role of mitochondria. Dr. Mahad, your project focuses on new understanding of progression in MS and a novel approach involving mitochondria that could lead to nerve protection in MS. In addition to describing your research, can you tell us what mitochondria are and why they are important in MS?
Sure. Thanks, Angela. First of all, it's very much been a collaborative effort, and very grateful to the Progressive MS Alliance for funding it and building things on top of the support we've received from the National MS Society and the UK MS Society. So Angela, in terms of the mitochondria, these are really the powerhouses of the cells. In our body, all different types of cells have these powerhouses, and they generate energy using the substrates that derive from the food that we eat, and it uses oxygen in a very efficient way to generate energy. So it's really an essential part of our body function, if you like. Now, the thing about the nervous system is, despite it being just about 2% of our body weight, it actually uses about 20% of all the energy. So the brain and the spinal cord is actually really fascinating. And during development, the myelin sheets, the protective sheets that are wrapped around the nerve fibers, actually makes this function from an energy point of view very efficient. Okay, so it's almost like you go to the gas station and you can get more miles for the gas that you're going to pay for, which is quite a lot of money these days. And that efficiency is made really through myelin. And of course, in MS, when the myelin gets damaged, you're kind of going back to the very early stages, if you like, of the development before myelination stage. And the brain is actually very robust, and it tries its best to try and reset itself for that energy consumption. So what we discovered is, basically, when the myelin is damaged in the nerve sheath, the nerve cell or the neuron tries its best to try and improve the energy function by generating more of the powerhouses, sending it along from the cell body into the damaged part of the axon. So this is actually just a natural process, and we call that armed, axonal response of mitochondria to demyelination. Bit of a mouthful, but armed. And what we discovered actually was that while it happens, it's the natural process, it isn't perfect. The timing of it takes a while, and you could really boost it using different genetic methods or therapeutics, pharmacology, and you can protect a lot of the nerve fibers, axons. So that basically means when the myelin is damaged, you can protect a lot more nerve fibers. So subsequently, repair of the myelin, like remyelination, can take care of the long-term neuroprotection. Okay, so that's essentially what we discovered.
Brilliant, Dr. Mahad. Thank you so very much. First of all, I'm excited to know that 2% of body weight, but it uses 20% of energy. That is phenomenal understanding. And outside of probably what you have discovered, but I'm thinking in terms of fatigue and
Right.
just the level of energy required by the body just to manage that could possibly explain MS fatigue. So thank you for that. It's my just way of thinking about things. So based on your findings, you and your team are exploring development of potential drugs that could protect the nerves of people with progressive MS. Can you tell us more about those drugs and how they could work potentially?
Yeah, so we took sort of a fairly focused way of selecting drugs. So this wasn't really an unbiased, comprehensive kind of screening method, right? We just went for what we thought might be the best candidates, and we tested about 6 different drugs and came up with one of the drugs that are used in diabetes called pioglitazone. And you have to be a little bit careful using that drug, because if patients have got heart failure and cardiac conditions, that can make it a lot worse. So you're not allowed to use that in those circumstances. But it is a drug that can potentially be repurposed. And we found this in multiple experimental systems actually to be quite robust in boosting that armed response. So in sort of cell culture models, brain slices in mouse models, this was highly effective, and it is actually fairly promising, because there's a NIH are doing a trial called TRAP MS using this drug. There's a couple of small studies that's been published, so something that we're quite keen. Obviously, we have to be realistic, and animal work translation into the humans. It's that there are issues with that, but it's something that we're sort of excited about.
I know I'm excited about these types of innovation and progress, and I'm sure others are as well. So let's now hear from Dr. Zalk, who is a distinguished researcher and the former director of the Research Center of the Brain and Spinal Cord Institute, now the Paris Brain Institute located in France. His scientific interest is the biology of myelin-forming cells in MS. Dr. Zalk, you and your team are working on discoveries that could result in remyelination in progressive MS. Your work is also very complex and involves microglia, the immune cells of the brain. I'd love for you to help us understand your project more and what you're learning about progressive MS.
Well, thank you very much, Angela. And thank you for the Progressive MS Alliance for supporting our research. If you would allow me, I like history. And to tell you about microglia, I will go back to the history, who discovered microglial cells. In fact, there is one very famous histologist named Ramon y Cajal. He described all the cells in the brain and spinal cord, with two exceptions. The oligodendrocyte, which is the myelin-forming cells in the central nervous system, and the microglia. And these two cells, which are the one which we are interested if we work in multiple sclerosis, he missed these two cells. And they were discovered by one of his students named Pio del Rio Ortega. And I just want to tell you a little story about the discovery. Pio had designed a new way of staining tissue section, and he could see these cells that had never been described before. And those were oligodendrocyte and microglia. But he did the experiment once, he tried to repeat it, couldn't see the cell. Repeated the second time, couldn't see it. Third time, didn't work. So he went back to his notebook, and he realized that the day the experiment had worked, that very day, the temperature was something above 32, 35, which would be in Fahrenheit, something like 95 degrees Fahrenheit, so very hot. So he realized that if it was so hot in the lab, he must have been using the fan to have some air. So he repeated the experiment with the fan, and he succeeded. And I like to tell the story because I think at least for the student, that you have to note everything on your notebook. Little details that you think are useless may be extremely important if you go back to your notebook. So that's for the discovery of these two cells, the microglia and the oligodendrocyte. Now, microglia are less known maybe than oligodendrocyte, and I call them the Janus of the brain. Janus is the Roman god that is always represented with two faces. On one side, the god is smiling, and on the other side, he's angry. During the Roman time, Janus was supposed to preside to the beginning of the conflict and to the end of the conflict, that is the war and the peace. And that's exactly what microglia are Dr. Jekyll and Mr. Hyde. They are involved in destroying the myelin sheath, the myelin which is around the axon, and that's why we want to fight these cells. But at the same time, just after, they will phagocyte all the debris and allow the remyelination. So that's what I call Dr. Jekyll and Mr. Hyde. You see, on one hand, they will destroy for reasons which we don't really know. But on the other hand, they are very helpful. And if they are not here, remyelination cannot start. And that's why I think it's important to study these cells. And what the project that has been funded by Progressive MS Alliance is just we are using two systems. One is a tadpole. It may look bizarre to work on tadpole, but they are very interesting animals. First, they are myelinated. Their myelin looks exactly the same as myelin in human, and we can have plenty of animals. So all our experiments, when you work on mouse, you have 10 or 20 animals with tadpoles. We can have 100. So, if we see something which is true in the tadpole, I'm confident that I can push it to other animal models. And what we are doing with Anne de Mazière, in fact, is to follow what these microglial cells are doing during the remyelination period. And what we have noticed is that these cells, they send processes all the time. And these processes are very active. And they touch the axon. They touch the axon not everywhere, but at the place where there will be what we call a node of Ranvier. The myelin is a sheet which is wrapped around the axon, but it's not continuously wrapped. It stops at what we call the node of Ranvier. And the microglia, they touch this area. What they are doing there, this is part of the project that Anne and myself are working on, and how often they will touch, how long they will maintain the touching of the stability of the touching. That's exactly what we are trying to do. Since these animals, the tadpoles, are transparent, so we can do that with live imaging. And that's then there is another part of our work which comfort me in the way that it is important to work on microglial cells, is that it is obvious that the cell that needs to do again myelin is the oligodendrocyte. We know that, and and Don has mentioned that we have also to put forward the energy to help these cells doing the work. What we found is that there are some molecules which are acting on microglia and which promote remyelination. And this was sort of not expected. And so this is one way we will continue our research to see to what extent these cells that you can push them to probably secrete, we don't know what they are doing, or to touch the axon at the place where there will be a myelin sheath, to what extent, what are they doing so that they will help the oligodendrocyte doing a new myelin sheath. So that's about our work on microglia. Maybe I was too long.
Not at all. I'm fascinated, particularly by the idea of using tadpoles. And I can just visually, I'm imagining this. And so being in the lab and being able to see that, I'd like to join you one day with that. So, but you're also exploring.
You're most welcome, and I will show you all my tadpoles.
Oh, thank you. You're also exploring compounds that could promote remyelination. Can you tell us about those efforts, what you're learning, and where we might go from here?
So I'm a medic by training. So I started to work on human. Then I moved slowly to rats, mice, and I've been working for many years also on chick and quail. And then I moved to the xenopus, this sort of frog. and the tadpoles, which are the baby of the frogs. And I think that going to more basic animal should help us. I'm not saying we'll find a solution, but at least we can have a lot of animals, we can find things, and then we can move it to more sophisticated animal closer from human. And really, you know, there has been several clinical trials that are involved in promoting remyelination. And so far, they all fail. And my reasoning is that because there was not enough functional testing, it was based on the histology, but we need to show that it's not enough to make myelin, but this myelin needs to be functional. And this is what we are trying to do with the tadpole. We are developing functional behavior tests, and we can then demyelinate the tadpole, and then we can see whether they recover their function. We test mostly the vision because it's more, for us, it's easier to test, and it's a relatively simple test. And we can test both molecules that will act on the oligodendrocyte, as I mentioned earlier. This is obvious because this is the cell that need to make myelin, but we can also test molecules that are working on the microglia and see whether they help doing a new myelin sheath, and that will be functional in the test that we are doing. One thing that I would like to add, speaking of progress, it's not only on tadpoles, but also because Paola mentioned the progress in MRI. So imaging in human is absolutely crucial. And we, with Bruno Stankov, have developed a sort of derivative of MRI, which is the PET, positron emission tomography. And this is, I think, the first test that can really follow the appearance of disappearance of the myelin sheath. It's really, MRI is always indirect, while the PET scan is direct, and we can, what Bruno has shown is that he can follow a patient during 4 months, and he can show that some patients are spontaneously remyelinating and some are bad remyelinator. And this is, I think, also very important to screen this type of patient, because we will have, you know, if you have a patient that never can remyelinate, then that's how you end up in a failure. So you should, you know, help the patient which are remyelinating, or the reverse. But I think it's very important to have a tool that will help us to see whether it is an efficient remyelination.
Thank you so much, Dr. Zhao and Dr. Mahad. And both of these are very exciting projects, and they provide so much hope, especially for those of us who are living with progressive MS. Yet these are only 2 of the 17 challenges in Progressive MS Awards. Dr. Zaratin, can you provide a brief overview and description of some of the other areas of research being accomplished?
Well, I think that the 2 examples we just heard about, I mean, it's amazing example on how can research work to understand the mechanisms that drive progression, and a very good example on how important is to understand mechanisms, because this will mean also to find marker to test potential therapy in vivo, as Dr. Zark just explained to us. So overall, this project are really working on understanding mechanism, and so we can speed up, accelerate the development of therapy for progressive MS. So in addition to the 2 areas of research we just heard about, there are other promising areas, but that are all linked together and linked to the topic we have just described. You know, it's about studying the role of oligodendrocyte function, studying myelin and myelin action interaction, and studying metabolites and cellular metabolism, because these are all linked, these mechanisms are all linked together. So just to give an example why oligodendrocyte is so important, and our expert just described this very well, but if you want to summarize this, I mean, it's a special glial cell that produce myelin sheath and also ensure the correct nerve conduction and provide energy into neurons. So if this is broken somewhere, then your car is not working anymore. It's not, and you need to understand how all these contributions work together. But the other area, important area, is fully dedicated to test molecule, as we just heard. So we have already some projects that are testing molecules on the above mechanism. And in addition, there are projects that are investigating markers of progression. So in addition to slow and reverse progression, we can, with this project, thinking to stop progression, because if we are able, through epigenetic studies such as the one included in the challenge our portfolio, to identify genetic factor, for example, we can even to recognize progression as early as possible, and thus be able to measure therapies over time.
I know that we will all be so anxious to hear what has been learned and how the findings will help advance the development of breakthrough treatments. For everyone watching, please know that we will highlight the results of these projects in a 2023 webcast. Dr. Zaritan, before we go though, do we have a rough timeline for when we will learn more about the results of all of these important projects?
So all our recipient will provide research progresses on mid-2023. And let's remind that these are projects that are developing or were developed under pandemic, so we should not forget this. In addition to this, as soon as we will have initial results out of challenge awards, Allianz plan to build on these results towards robust projects that will translate finding to move along a specific development pathway for translational and clinical trials. So we do hope that we can really build on these results. But the important things is that we expect that these challenge awards will provide really innovative findings and able to shift the paradigm. And another important aspect is people with MS, as in overall Allianz portfolio, will be engaged in understanding the results with us and understanding what is priority for them and how we should build on these results.
Thank you so very much. Well, we're nearing the end of our time today, but I would like to ask each of our MS experts one final question. People affected by progressive MS may be easily discouraged by the lack of progress in solving progressive MS, but that finally seems to be changing. What would you want people to know about progressive MS research? Dr. Mahad, let's begin with you. And you're on mute.
Sorry, as we know, there's been many advances in the relapsing form of MS in terms of treatment, but having even one approved therapy for secondary progressive MS and one for primary progressive actually changes the clinical landscape. Before you tried not to diagnose patients too soon, and you thought, oh, what am I going to do? Now there's actually a positive motivational factor to recognizing it early, and being able to try and target that early is going to offer a huge potential rather than just wait for. I mean, that alone I think is a massive change in our sort of approach to progressive MS. And plus, of course, that's going to focus our research attention, like we've discussed, in terms of understanding fundamental mechanisms, how you can target it and translate it, and developing clinical platforms. So I think we probably also need to develop some of the clinical platforms to try and target progressive MS early and recognize, there's a lot of imaging. So I think this is a fast moving field, and younger generations of researchers are coming in, inspired by Progressive MS Alliance and the collaborative effort. I think there's so many things are lining up.
I'm very encouraged and very heartened to hear the connection of research to clinical clinical work. I'm having an MS moment right now. I can't think of the word, but the whole clinical treatment plans. And so being able to actually take it from research to the clinic, the patient, and having us all involved, I think this is phenomenal. So thank you very much for that. Dr. Zalk, what would you tell people about research in progressive MS?
Again, I will go to history. MS was first described in 1868. And for 100 years, there was no progress. There was no hope. I mean, we are giving cortisol to the people. Then in 1995, they introduced the beta interferon. And I'm not saying that beta interferon is curing the patient, but it has been a fantastic progress. Now there are 12 or 15 drugs which can already ameliorate the quality of life of patients with MS. So I cannot say in how long it will take now for progressive MS patient to find a solution or maybe different type of solution. But I'm quite optimistic because there has been quite amount of money that has been put into this type of research. And I'm confident that we will end up with something. I cannot tell exactly. As Don had said, progress in the platform, progress in biomarkers. That is everything that will help defining more precisely which type of patient you have. Then because if you take everything, then you I don't find the word in English, but you need to have something more precise for each patient so that when you try something, it will be directed to this one. And I think we are going into this direction. And I'm quite positive about the fact that I don't know how long it will take. But again, for relapsing remitting, it started in 1995. It's not such a long time ago. It's only 30 years ago. And it has been a real improvement for the patient. So I hope it will be the same for progressive MS. That's it.
Thank you so much. And I'm really intrigued by the idea that we can move towards a more individualized treatment plan for individuals living with MS. But that is going to involve the collaborative effort of multiple researchers in multiple areas. And so, again, thank you so much for your input and your insight into this and your love of history. And so, Dr. Zaritan, what would you add to these comments?
Well, it's easier. I mean, I think stronger, together we are stronger. I think that talking about the alliance efforts, I do believe that, collaborative effort will provide a significant progresses to our effort to find a therapy for progressive MS. And this is also a scientific basis. I mean, there is a hidden part of the MS iceberg, that is made up of different mechanisms, of different aspect factor. So, and to make it in terms of being able, as Dr. Zalk said, to find a personalized treatment, we need to look at this all factor together in a holistic manner. And last but not least, capturing people experiential knowledge and make it scientifically relevant, as you well know, Angela, will make a huge difference also for progressive MS.
Yes, thank you so very much. Well, today we examine some of the leading questions regarding treatment and research in progressive MS. As I hope you have experienced by today's panelists, the Alliance has the very best people in the world working every day to find solutions to the challenges people affected by progressive MS face. The Alliance continues to inspire hope and achieve progress. Thank you to our MS experts, Dr. Zaratin, Dr. Mahad, and Dr. Zalk. And thank you to everyone watching who joined us today. Please stay connected to the Alliance through our regular webcast and your MS organization. And please share the work of the Alliance with others. We need everyone to be a part of this worldwide effort. Together, we are stronger than progressive MS.